Okay, so you've probably heard about CRISPR, right? And how it's like totally changed the game in genetics.
Huge revolution.
But what about the stuff inside the cell, the sort of dark matter, the mitochondrial DNA, in those tiny powerhouses?
Yeah, the mitochondria.
Editing that has always been, well, almost impossible.
That's right.
Until now, maybe. Today, we're diving into a breakthrough that could change everything on that front.
It's really fascinating stuff. And, the reason it's been such a challenge is because mitochondria are these unique power plants in our cells.
Right, they make the energy.
Exactly. They take the food you eat, the oxygen you breathe, and convert it into ATP, the chemical energy cells need.
Yeah.
But the critical part here is they have their own DNA.
Separate from the main stuff in the nucleus.
Completely separate. A small circle of DNA, mtDNA, and you get it almost entirely from your mother.
Okay.
And that distinct nature, its location, its structure, it's presented this massive hurdle for standard gene editing tools.
So that's our mission for this deep dive. We want to unpack this new science, this potential way to edit mitochondrial DNA. We'll look at what it could mean, maybe, for treating some rare inherited diseases, and also touch on the links to aging, what's real, what's hype.
Yeah, important distinctions there.
By the end, you should have a really solid grasp of what this breakthrough is, and just as importantly, what it isn't yet.
Sound good?
So, let's set the scene. CRISPR, as we said, amazing tool. Scientists use it all the time for nuclear DNA. But why the blind spot for mitochondria? Why has it been like this impenetrable fortress?
Well, the standard CRISPR system, CRISPR-Cas9 usually, relies on a guide RNA. Think of it like a GPS molecule that tells the Cas9 scissors exactly where to cut the DNA. The problem is getting that guide RNA across the two membranes surrounding the mitochondria. It just doesn't happen easily.
So the delivery system fails.
Pretty much. The doors are locked, basically. So direct, precise editing of mtDNA using that standard CRISPR toolkit. It's been incredibly difficult, almost a non-starter for a long time.
Right, until this new work.
Exactly. This is where the new approach comes in, what the researchers developed. They're called CRISPR-free base editors.
Okay, CRISPR-free, so different mechanism entirely.
Totally different. And this is the clever part. Instead of cutting the DNA double helix, which is what Cas9 does, these base editors perform a chemical conversion. They directly change one DNA letter, one base, into another. So, for instance, they can precisely swap a C, cytosine, to a T, thymine, at a very specific location. No cutting involved.
Wow, okay. So it's more like chemical surgery than molecular scissors.
That's a good way to put it, yeah. And then there's the how do they get it in?
Yeah, the locked doors.
So for that, they use something you've probably heard about recently, lipid nanoparticles, LNPs.
Like from the COVID vaccines, the mRNA ones?
Exactly the same principle.
Yeah.
Tiny little bubbles of fat that can fuse with cell membranes and deliver cargo inside. In this case, the cargo was the base editor machinery. And using this method in mouse cells in the lab, they successfully delivered the editors into the mitochondria and made those precise C to T changes in the mtDNA.
That's pretty huge.
It really is. If you connect this to the bigger picture, overcoming that delivery barrier and using a non-cutting editing method, it sidesteps the old problems in a really elegant way.
Yeah.
It opens up a pathway we just didn't have before.
Okay, so they found a potential key, a way in using these base editors and LNPs. Assuming this holds up, gets refined, proves safe down the line. What does this actually mean for people?
Well, the most direct potential application is for certain rare inherited diseases caused by specific mutations in the mitochondrial DNA.
Like what kinds of diseases?
Things like certain forms of mitochondrial myopathy, which causes muscle weakness and fatigue, or Leber's hereditary optic neuropathy, a type of inherited blindness. There are others too.
So conditions caused by just one or a few specific typos in that mtDNA.
Exactly. If you know the specific mutation causing the disease, this kind of precise base editing tool, in theory, could one day be used to correct it.
Right.
It potentially makes these incredibly difficult conditions more treatable at their root cause.
Okay, here's where it gets really interesting for a lot of people listening, I bet. What about aging? We hear a lot about mitochondrial health and longevity. Does fixing mtDNA errors touch on that?
Right. So there's definitely a known link. Mitochondrial function does decline with age, and damage to mtDNA accumulates.
Yeah.
This dysfunction is implicated in various aspects of the aging process.
So could fixing that damage slow aging?
Well, that's the big question, and where we need to be really careful. Some researchers speculate that if you could reduce or repair that age-related mtDNA damage, maybe you could influence age-related decline.
What?
And this is a big, but that idea is still firmly in the realm of hypothesis. It is absolutely not established science. We're nowhere near proving that connection, let alone developing therapies based on it.
Okay, really important clarification. Thank you. So let's hammer that home. This research does not mean we're about to reverse aging.
Absolutely not, no.
And it's also not a magic bullet for all mitochondrial diseases either, right?
Correct. Many mitochondrial disorders are incredibly complex. They might involve multiple mtDNA mutations, or the problem might actually lie in the nuclear DNA, the genes in the cell nucleus that code for proteins needed by the mitochondria.
So it's a complicated picture.
Very. These base editors are precise tools for specific mtDNA targets. They won't fix everything, even within the realm of mitochondrial problems. The genetic landscape is complex.
And besides the complexity, there's the stage this is at. You mentioned mouse cells in a dish.
Exactly. This is foundational research. It's currently at the cell culture stage. It hasn't even been tested in whole animals yet, let alone humans.
Right.
And the path from lab discovery to an approved therapy that you or I could actually receive, that's typically a decade. Minimum. Often much longer. There are many, many steps involving safety, efficacy, animal models, human trials.
So it does raise that important question then. If it's so early, so many hurdles, why is this still seen as such a significant development?
That's a fair question.
Because progress here in editing mtDNA specifically has really lagged behind editing the nuclear genome. For years, it felt like this area was just stuck.
It really did.
So seeing this kind of cautious, methodical, technically really impressive work succeed, even in cells, it feels like a meaningful step forward. It's like, okay, that blind spot we talked about, maybe the fog is starting to lift.
I think that's exactly why it's significant. It's a proof of principle.
Right.
It shows that targeting and editing mtDNA is feasible with the right approach.
Right.
And if, again, it's an if, but if this eventually leads to safe and targeted therapies, it could genuinely make a difference for people with some really devastating, currently untreatable genetic conditions. That potential is huge.
Okay. But there are still big challenges ahead, you mentioned.
Oh, absolutely. Many challenges remain. We need to ensure these edits are truly specific and don't cause unintended changes elsewhere, what we call off-target effects. Long-term safety is paramount.
And delivery, getting it to the right place in the body.
That's another major one.
Yeah.
How do you deliver these editors efficiently to the specific tissues affected by a particular disease, muscle, eye, brain, and not everywhere else? That's tough.
Yeah.
Then there's scaling up manufacturing for clinical use, the cost, and of course the ethical considerations that always accompany powerful gene editing technology.
Right.
But, this is how progress often happens in science. It's steady, it's careful, it's incremental. This isn't hype. It's responsible research addressing a real need, building a foundation, and it holds genuine long-term promise.
So let's bring it together then. We've looked at what is undeniably a really significant scientific step, tackling a notoriously tricky area, editing mitochondrial DNA. And while we've stressed this isn't some instant cure, it's not reversing aging tomorrow, it really does open up some profound possibilities for treating very specific, very difficult genetic diseases down the road.
It absolutely does. And it makes you think, doesn't it? Even at this early stage, how does this change our basic approach? We now have a potential tool to interact with this whole other genome within our cells.
Yeah, that's true.
How might having this new access, this ability to potentially read and write mtDNA more effectively, how might that spark completely new ideas, new lines of research into base goal biology, maybe even into aging itself eventually?
That raises an important question indeed.
What surprising connections that we uncover now that we can probe these tiny powerhouses, these mitochondrial instructions, in ways we couldn't before. It really opens up the field.